Thank you, ma'am. Good afternoon to all of you. I am Ajay Kumar Patel. I did my PhD from Indian Institute of Technology, Roorkee, India. And thanks for invitation. And thank you, Professor, for my given this opportunity. And in this presentation, I will talk about the evolution of soil fertility status through mixed pixels analysis of hyperspectral data. So in this presentation, I will cover the introduction part of the topic, then literature review, so research gap, objective of research work, then background theory, samples and data, methodology, results and discussion, and followed by the validation conclusion. So let's start introduction. So the fertility is necessary for plant nutrition to maintain the soil fertility. And as we know, the appropriate type and quantity of nutrients enhance the quality, yield, production. But the inadequacy and unreasonable nutrients creates problems such as unhealthy crops, increased input cost, poor yield, and environmental pollutions. So thus there is a need for the assessment of the soil fertility status, and these are the primarily two types: destructive and non-destructive ways. So in destructive methods, these type of methods are quite laborious, complicated, lengthy, and costly to operate, and also this is not environmental friendly. So and non-destructive ways capable enough for on the go in situ sensing and assessment of the soil properties. These methods help to increase crop productivity while it mitigate detrimental environmental effects. Therefore, many researchers have sought real-time, non-destructive methods for soil fertility status assessment. So, non-destructive methods, as based on the sensors and data use, they are basically divided in four types, like optical radiometric sensor, which is based on the absorptions, reflectance, and or emitted emissions of the electromagnetic radiation. And second type sensors based on the electrical electromagnetic, which is the accumulate or conduct electrical charges. Third type of sensor, electrochemical sensors, which uses the works with the release ions of the sample. And fourth one is the mechanical, which is the resist mechanical distortions. Past few years, frequently hyperspectral imaging technique has been utilized for assessment of the soil fertility status. It contains both spatial and spectral information and capable enough to quantitatively assess the chemical compositions of soil samples. So, in hyperspectral, which contains many continuous spectral bands, and it provides an abundance of spectral information. Hyperspectral is a very effective technique to analyze the composition of each material, and due to the fact that different materials poses different properties under different electromagnetic spectrum. Hyperspectral are particularly suited for in situ measurements. It can lower the required number of field samples and eliminate the need of chemical reagents. It reduces the time and cost implications associated with field data collection and analysis. The soil fertility status can be analyzed in real time over a larger spatial area using hyperspectral technique. This attracted the attention of a wide range of recent agriculture studies and advocating for its continued use for precision agriculture. In the literature review, many researchers, many authors have been worked using hyperspectral remote sensing for the assessment of the soil fertility status, and there are many kinds of ways they have used. Like there is some authors have used the vegetation indices. Many authors use some particular wavelength region to identify the soil fertility, and many authors using both spectral and multispectral and hyperspectral data set to analyze the soil fertility status. And some authors also uses the artificial neural network, vector machine based classification methods, and they uses satellite data as well as airborne data as well as spectroradiometer field data also. And These authors are capable of satellite-based hyperspectral vegetation indices for estimating soil salinity in agriculture fields, evaluate the performance of 21 existing vegetation indices, and developed a new vegetation indices based on a combination of wavelength, sensitive, or multiple stresses, and find the best one of estimating soil salinity. Recently, by 2018, mapped the soil alkalinity and salinity using hyperspectral technique. And 2019, Zhang investigated the prediction of soil salinity with soil reflected spectra. 2020, Milwaukee have been examined the advantages of hyperspectral imaging for the assessment of soil degradation due to salinity. So HSI has been employed for the identification and determination of soil nutrients properties through the analysis of spectral absorption features in recent decades. Many researchers have adopted this technique, specific wavelength region, to distinguish the soil variety, analysis of soil polluted with hydrocarbons for soil salinity characterization, prediction of the presence of topsoil nitrogen, and recently 2019 and 2020, many authors have been utilized the non-destructive capabilities of hyperspectral imaging to determine the nitrogen fertilization levels within the soil. Advantage of these methods is that authors perform qualitative analysis, but they have not provided quantitative analysis of salt-affected soil and micronutrients in different types of soil. So the research gap through literature review following research gaps in the assessment of soil fertility status using hyperspectral imaging has been found, like in analysis of salt-affected soil and soil micronutrients with estimation of their fractional abundances using non-destructive techniques. Like in situ assessment of soil fertility status through mixed pixels analysis of hyperspectral data require enhancement of both the accuracy and computational speed for estimation of fractional abundances of nutrients in soil. So the objective of this work is for the assessment of the soil fertility status through mixed pixels analysis of hyperspectral data. The study intends to introduce a method of integration of spectral derivative and a linear mixing model termed as derivative analysis for spectral unmixing called DASU, and that followed by deep learning technique, a novel approach. The objective has been realized through applying DASU for estimation of fractional abundances, quantitative analysis of salt-affected soil and micronutrients in soil. Applying a DASU-based deep learning network to enhance both the accuracy and computational speed of the spectral unmixing process for computation of fractional abundances of urea fertilizer in soil samples. In the background theory, multiple ground objects coexist in a single pixel of the hyperspectral data that is called mixed pixel, and these are frequent in remotely sensed hyperspectral images due to the insufficient of spatial resolution of the imaging spectrometer or due to the intimate mixing effect. Mixed pixel basically are occur because of two types there happen, that is a microscopic level, and this is the and second is the microscopic level. These based on the type of the mixing ratio and type of the contents or the objects of the pixel. So if the objects are very close, very intimate, then this kind of mixing is called microscopic, and there is a sort of big size of the sample, and that is called this kind of mixing called the microscopic mixing ratio. In spectral mixing is an essential step for categorizing the mixed pixels within hyperspectral data. In this process, the measured spectra of a mixed pixel is decomposed into products of different end members, that is called pure pixel, and their corresponding abundances coefficients, that is the mixing ratio. Spectral mixing methods are categorized into linear and non-linear mixing model. These are the based on the type of the scattering occur during the data acquisition. Steps for spectral mixing, basically 3 steps to involve for spectral mixing of hyperspectral data, which is the estimation of end-member numbers, means how many pure pixels are the present in our data set, then end-member extraction, the type of the end-member extraction, and third one is the abundance estimation when the means the mixing ratio. So this research work is based on the estimation of fractional abundances in the mixing ratio of a mixed pixel of an object of an hyperspectral data for spectral mixing. Derivative spectroscopy and continuum removal techniques can be used to enhance the absorption features in spectral reflectance curves of hyperspectral data. Therefore, spectral feature analysis approach based on the linear mixing model are used for spectral mixing. So in background theory, we have utilized first spectral derivative. This involves finite divided difference approximation to compute the change in spectral reflectance curve over a bandwidth, and without any loss, we obscenity, we assume lambda L is greater than lambda K. Both are the corresponding wavelength regions of a band. And then I have utilized continuum removal. Continuum removal is a normalization approach. This could emphasize the location and depth of individual absorption features, drawing a curve with the absorption values from 0 to 1. That is the range. And it provides more causal relationship between reflectance features and the corresponding amount of absorbing material. And this is the mathematical representation of the continuum removal technique. Then I have utilized the linear mixing model. The reflectance of a pixel in each spectral band is expressed as a linear combination of the characteristic spectra, and thus the reflectance spectra of mixed pixel can be explained like this mathematical equation, where there is the mixed pixel spectra, and this is the multiplication relation of the abundance of the mixed pixel and the pure pixel into member of the data set of the pixel. And we assume that its spectral derivative will also comply to the linear mixing model, and then equation third can be expressed as the after taking the derivative of the linear mixing model, and the constant can be satisfied here. The value of the fractional abundances must be between the 0 and 1 or equivalent. And then deep learning network, which I have utilized in this work, huge volume of hyperspectral data can be difficult to analyze and process using conventional methods. So therefore, a relevant information may be extracted from this complex dataset using artificial neural network. And these are the most important network which has been designed according to the neural structure of the human brain. Artificial neural networks are used to extract deeper features and association between various inputs by the use of hidden computational layers. These have two or more of hidden layers are known as deep neural networks, and indicating the large number of layers used. So and the validation after using the proposed methodology, in the last I have used the validation methods, which is the based on the laboratory examination, and these are the destructive methods. So for electrical conductivity and pH for computer to measure the salinity in soil, and oil chromatograph was used for determining the concentration of nitrogen in the soil sample. Then stannous chloride method was used for determining the concentration of phosphorus, and flame photometer was used for determining the concentration of potassium in the soil sample. So this is the study area where I have collected our soil sample. There are three types of soil samples. I have collected silt clay type of soil, sandy and loamy. This is collected from the Roorkee in September 10th, 2018 at 12:30 p.m. During the collection, I have also used a GPS device, and using GPS, I have stored these latitude and longitudes. And during the sample collection, there was the 21 degrees Celsius temperature, and humidity was approximately 61%. Soil micronutrients are mostly concentrated on the surface of the earth. Therefore, samples were collected within the soil horizon at a depth of 15 to 20 centimeter. Approximately 5 kilograms of each soil tap was collected in a polythene bag, packed, labeled, and brought into the lab for further analysis. The soil samples were dried in an oven at 105 degrees Celsius for 24 hours, taken after grinding. For removing the impurities of the study soil samples, 2 mm sieve was used for the sieving. So in first study sample, in this study, I have used three types of data samples. So in first study samples, we have used sandy soils and 5 pure salt minerals. And I have prepared 15 mixed samples, 3 for each salt mineral, but using the different compositions. So these are the pure samples, salt samples, and this is the soil, pure soil, sandy soil, and these are the boxes which I have used during preparation for the mixed samples. So 1.5 centimeter thick soil is considered as a optically finite soil as per the literature, and therefore all prepared 15 mixed and one controlled soil samples were kept in a total of 16 top open rectangular plastic boxes with a profundity of 3 centimeter. Each plastic box was filled with 100 gram of sandy soil, and the saline solutions were prepared by 4 gram volume of each salt that was dissolved into 200 ml of distilled water using standard methods applied in laboratories. Saline solutions were separately poured into 100 gram of soil samples with volume of 25 milliliter, 50 milliliter, and 100 milliliter solutions. Based on the different levels of soil salinity, 3 mixed samples of each salt were artificially prepared. One sample was treated with distilled water as a controlled soil sample. The sample were placed in a bigger plastic boxes in such a way that added salt solution thoroughly mixed with soil. The prepared samples were left to dry at room temperature. In second type of study, I have used three types of soil: silt clay soil, sandy soil, and loamy type of soil, and in NPK compost, which having 19 ratio of the nitrogen, phosphorus, and potassium. And 15 each mixed samples were prepared separately with different compositions. And as shown in figure, I have prepared these mixers like this. So to ensure that the NPK compost is thoroughly mixed within the sample, it was stored for 4 days. The contribution of each macronutrient in the sample was computed by a conversion factor calculated using their respective molecular mass. Among all prepared samples, 4 pure samples are used for qualitative analysis. 45 mixed samples were used to quantify the percentage of soil and NPK compositions in the sample. In third type of soil study samples, these samples we have used two soil types, loamy and silt clay, and urea fertilizer, and prepared 15 mixed samples with different compositions. Source and characteristics of hyperspectral data. A portable spectroradiometer was used in laboratory for the acquisition of hyperspectral data of all study samples. It covers the wavelength range of 350 to 2500 nanometer with 994 continuous spectral channels. The processing of recorded hyperspectral data were performed by using the SPC Spectro Vista Corporation XHR 1024i Personal Computer Data Acquisition Software version 1.19.3. So the methodology, in the part of methodology, soil samples were collected into a polythene bag, labeled, sealed, and taken to the laboratory. During collection, there was a 14% approximate moisture. Drying of the sample was done in an oven at a temperature of 105 degrees Celsius for a 24 hour, followed by a hand cleaning and grinding. Sieving of the sample was carried out by a 2 mm sieve, grinding 5 pure salt minerals. Those are halite. These are the salts, and was used and purchased from the high media laboratories. Grind commercial inorganic NPK compost and urea fertilizer was used. Salt minerals and fertilizers addition in study soil samples was done for the purpose of preparation of mixed samples. The spectral reflectance curves of all study samples was recorded using spectroradiometer in the laboratory. Continuum removal and spectral derivative technique were employed for computing the absorption features of measured spectral reflectance curves. Based on the characteristics of the spectral absorption features, the optimal wavelength was selected. For estimation of fractional abundances in soil samples, an integration of spectral derivative and linear mixing model called DASU has been used. DASU, followed by a deep learning algorithm, has been utilized to enhance the accuracy and computational speed of estimation of fractional abundances in soil samples. To train the deep learning network, 10,000 mixed pixels spectral response curve of study samples were generated using the heuristic distribution. Accuracy assessment of all results was performed using the regression models and coefficient of determination. For validation of the computed results, laboratory-based chemical analysis method was used in each study. This is the flow chart of my research work which I have done. So now I will discuss about the results and discussion. So starting from the study 1, fractional affordances, study of salt affected soil. So spectral, as shown in figure, spectral reflectance curves of 16 end members, and 1 for sandy soil and 5 for salt minerals. So total 150 scans of mixed SRCs were recorded for compositions of sandy soil and 5 salt minerals separately. Based on the salinity level, the average of 10 scans per mixed sample was stored and considered as the final SRCs. Gradual decrease in soil reflectance with increasing salt content is clearly shown in the figure. Mainly as greater than the wavelength region greater than to 1400 nanometer. And then I have calculated the continuum removal reflectance of each study samples per measured, and this is the plot of that data sets. Then continuum removal reflectance spectra of soil treated with the 5 different salt mineral solutions are shown in the figure, and representing 3 levels of salinity. For diagnostic absorption features, approximately near 1000, 1400, 1900, and 2200 nanometer are pronounced as salinity level increases. These are the wavelength region. Some other slight absorption features near 1100, 1800, and 2400 nanometer also can be identified from continuum driven reflectance. These absorption features also exist in the spectral reflectance curve of pure study samples. The absorption at 1000, 1100, and 1800 nanometer were caused by hydroxyl ion and water within the lattice of hydrated minerals. Deeper absorption features near 1400 and 1900 nanometer were probably the combined results of OH stretches and HOH bending fundamental sand over tone. Features at around 1451 and 1952 nanometer are weaker in the soil spectra, and their positions shift as the amount of the salt abundances in the soil decreases. This is due to the fact that soil absorption features around 1400 and 1900 nanometer broaden as the salt concentration in soil increases. Depth of these diagnostic absorption features varies with the degree of soil salinization and may be used to estimate the fractional abundances of salt affected soil using hyperspectral data. And then after continuum removal, I have calculated the second derivative results of spectral reflectance curves of soil treated with 5 different salt mineral solutions are shown in this figure, and representing 3 levels of salinity. So second derivative results of 4 pure salt minerals shows strong absorption features at 1899.2 nanometer, while viscophyte salt minerals shows weak absorption features because they contain more water content, and reflectance spectra of non-saline soil showed weaker second derivative values at 1899.2 nanometer, and these values increase as salt concentration in soil increases at this wavelength region. Therefore, it was easy to distinguish the differences among slightly, moderately, very, and non-saline soil. And however, spectral curve of very and moderately saline soils had some overlap sections but could be discriminated approximately from 1900 to 2050 nanometer wavelength regions. Box plots were evaluated for the spectral derivative outcome of the 10 pure spectral reflectance curve for each study end members as shown in figure. Box plots of spectral derivative values revealed that SRCs of all study end members dominated at 1899.2 nanometer wavelength region, and based on the above mentioned findings, the spectral absorption features at this wavelength region was selected for estimation of the abundance of mixed study samples. So therefore, based on the our previous results, 1899.2 nanometer wavelength region was used to estimate the fractional evidences using the equation 4. And based on the salinity, these were divided into 3 levels, like slightly, moderate, and very saline samples. The salient point of the observed result is DASU estimated of fractional abundances of mixed study samples give a better result at 1899.2 nanometer spectral region. Overall evaluation of DASU estimated abundances of mixed study samples was prepared through a box plot as shown in figure. So based on this study, I have chosen a second derivative equation, which is the wavelength region of 1899.2 nanometer. Now the accuracy assessment was done through laboratory-based measurement of electrical conductivity and pH. Calculated pH values of the study samples were ranged from 7.03 to 8.61 with an average of 7.64. Computed electrical conductivity of study samples were ranged from 2.51 ds per m to 15.2 ds per m with an average of 7.17 ds per m. Soil with a pH value 7.5 and electrical conductivity above 4 ds is considered alkaline and salinity respectively. So increment of electrical conductivity and pH values in mixed study samples means of increasing salinity level in salt affected soil samples. The average values of electrical conductivity and pH indicate that study samples are predominantly saline. Based on the results of laboratory based measurements, it is found that DASU approach performs well for estimation of fractional abundances of salt affected soil at 1899.2 nanometer wavelength region. And this is the table, the results of DASU estimated fractional abundances and laboratory test, which is electrical conductivity and pH of study samples. So these are the samples for one kind of salt. I have taken 3 kind of mixtures, and this is the electrical conductivity values. This is the pH values, and based on the results, I have divided the salinity level, slightly saline, moderate saline, very saline, and these are the results which I have obtained using the DASU method approach. Now in the second study, fractional abundances of a study of macronutrients in soil, total of 4 end members, spectral reflectance curves, where 3 for different soil types, and 1 for NPK compost was recorded. Total 450 mixed spectral reflectance curve were stored for compositions of all soil types, and an average of 10 scans per sample in order to minimize the instrumental noise was considered as the final reflectance. So this is the pure reflectance of the 3 soil types and 1 NPK compost, and these are the mixed reflectances of the each study samples. And then I have calculated the second derivative values of the all samples or pure samples. And since the spectral regions 989.3 nanometer and 1899.2 nanometer are associated with the amide NH functional group, therefore the computed second derivative spectral values of pure NPK compost at these spectral regions shows high positive peak, while the second derivative spectral values of each soil type show negative as well as less positive peak values. Meanwhile, at 2195.1 nanometer and at 146.9 nanometer, second derivative spectral values of all soil types show high positive peaks because of clay minerals, while second derivative spectral values of NPK compost at this wavelength region display the nearly linear spectral shape. So also to verify this conclusion, we have used the box plot, and in the box plot also they confirm these wavelength region suitable for 2 wavelength region is suitable for the NPK compost, and 2 wavelength region is suitable for the soil samples. And based on the above observations, spectral absorption features of 989.3 nanometer and 1899.2 nanometer was included for the estimation of abundances of NPK compost, and 2195.1 nanometer and 2346.9 nanometer was spectral region was used for estimation of abundances of each soil type, and these wavelength region in these wavelength region, we have I have used equation 4 to estimate the fractional abundances of the NPK and soil samples. And these are the observations which I have calculated using the equation 4. So these are the true abundances values. This is the mixture 1, mixture 2, mixture 3. These are the samples which I have prepared, 15 mixing samples using the soil samples. This is NPK sample, and this is for the clay soil and the NPK samples. So we have prepared 15 mixed samples, and these are the true evidences, means having 99% soil, some second mixture having 98% soil, 97%, 96, 95, 93, 92. These are the true evidences, and these are the true evidences, ratio of the soil and ratio of the NPK, and these are the estimated abundances in the wavelength region 989.3 nanometer, and these are the wavelength region estimated samples of this wavelength region. And we have calculated the error using the subtracting the true abundances and the estimated abundances. And same, this is for the second type of soil, for the sandy soil sample, and third one is for the loamy soil samples. These results I have calculated for each soil sample. Then the salient points of the observed results from table 2, table 10 shows that DASU estimated abundances of NPK compost gives better result at 989.3 nanometer wavelength region as compared to 1899.2 nanometer spectral region, and same in the case of our soil gives better result for 21.9.95.1 nanometer spectral region as compared to 2346.9 nanometer spectral region. So the correlation between the true and DASU estimated abundances were examined using the linear regression statistics as at a 95 confidence interval. So in this plot, I have this is the linear regression plot for estimated and true abundances and estimated abundances, and we have find out the coefficient R square values, and based on these we have concluded that two wavelength regions are suitable. 989.3 nanometer is suitable for NPK compost, and 2195.1 nanometer wavelength region is suitable for the soil composition. And these observations, this is also shown in the we have shown using the box plot. Statistics of box plot shows that absolute error are less in case of compositions of NPK compost at this wavelength region as compared to 1899.2 nanometer wavelength region. And similarly, the absolute error are less in case of compositions of each soil types at 2195.1 nanometer as compared to 2346.9 nanometer, as shown in the box plot. So as per conclusion, I have chosen two wavelength region, second derivative values of the any mixed sample, and mixed sample which the NPK compost, and mixed sample having mixed with the soil sample at this wavelength region. So we can easily estimate the fractional abundances values of NPK having mixed with soil, or soil sample mixed with NPK at this wavelength region. And the validation purpose, laboratory-based chemical analysis of study soil samples were carried out using standard methods. It aims to evaluate the availability of soil micronutrients, nitrogen, phosphorus, potassium in prepared mixed samples. Computed 21.14% nitrogen instead of 19%, 16% phosphorus pentoxide instead of 19%, and 17.5% potassium oxide from the pure NPK compost sampling gram per 100 gram. Amount of micronutrients present in compositions of soil and NPK compost was observed individually from 4 mixed samples as well as computed through conversion factors. Overall comparative evaluation between the DASU estimated fractional abundances and chemical analysis results of soil micronutrients are shown in this table. So this is the table where we have compared the DASU and laboratory test chemical analysis results of abundances of individual nutrients intubated with the mixture of soil and NPK compost. So the results of the chemical analysis shows that nitrogen observed from the mixed samples are comparatively less than nitrogen calculated by conversion factor. Observed phosphorus and potassium from the standard methods are close to the values calculated by the conversion factor. Based on the results of chemical analysis, it is found that the adopted Darcy approach performs well for estimation of fractional abundances of compositions of three soil types at NPK compost at spectral regions of 2195.1 nanometer wavelength region. And now in third study, estimation of fractional abundances of nitrogen in soil, spectral reflectance curve of three members, two for each soil type and one for urea fertilizer was recorded. Spectral flow reflectance curve were acquired for prepared mixed samples of each soil type in 11 replicates, totaling to 330 scans. Two sets of abundances were generated from distillate distributions, one for the combination of urea fertilizer and pure silty clay soil, and second for the combination of the pure loamy soil and urea fertilizer. A total number of 10,000 each mixed spectral reflectance curve for both the data sets were generated to evaluate the performance of a deep learning network. So this is the plot of the pure samples, two pure soil samples, and this is the reflectance curve of the urea, and these are the mixed spectral reflectance curve of the two types of mixed samples, one having loamy soil and one having with the sandy soil, and these are the mixed 10,000 mixed spectral reflectance curves are generated using the distillate distribution, one for each soil type. Then I have calculated spectral derivative values. Spectral derivative results for pure urea fertilizer shows high positive peaks at 9893.3 nanometer and 1899.2 nanometer wavelength regions, whereas the spectral derivative results of soil appears negative with these wavelength regions. Similarly, the soil types display positive high peaks at 2195.1 nanometer and 2346.9 nanometer spectral regions. On the contrary, the spectral derivative results of urea fertilizer shows an almost linear shape, thus the derivative values are closer to zero at these wavelength region in the case of urea. Then I have calculated the Continuum removal spectra to identify and isolate the characteristics, absorption features of micronutrients, minerals, and water in soil. The output of Continuum removal spectra for both the soil types reveal the high absorption peaks near 1400, 1900, 2200, and 2350 nanometer. Similarly, the Continuum removal spectra of pure urea fertilizer generates high absorption features near 1000, 1500, and 1500 and 1950 nanometer spectral regions. Then we have also calculated the box plots for these wavelength regions, and SRC of urea fertilizer dominates at 989.3 nanometer and 1899 nanometer wavelength regions, and for in the soil case 2195.1 nanometer and 2346 nanometer wavelength region is dominated in the case of soil. So as per the above findings, spectral absorption features for this wavelength for these two wavelength regions I have utilized for the estimation of the abundance of urea fertilizer, and two wavelength regions are utilized for the evaluation of the abundances of soils, and using the equation 4, that is the linear mixing model, after the taking the derivative of the linear mixing model equation. So, and the correlation of these results have been done using the linear regression model. So this is the plot for each wavelength region, and based on the coefficient results of the R square, there is a we found that the wavelength region 1819 Dasu computed abundances for all inverts yield at least variation at 1899.2 nanometer for urea fertilizer and 2195.1 nanometer for both soil type as compared to the actual abundances. So Dasu computed abundances yield relatively higher differences at nanometer for urea fertilizer and 20 nanometer for both soil types as compared to the actual abundances. So these are the results of the R square values. So based on their we have find out these two wavelength region, and then also I have used the DASU based deep learning network was used for estimation of abundances. This is the next step for our approach. The DASU estimated abundances were used to train the deep learning network, and deep learning network contains one input layer with 994 nodes. This is the input layer, and we have used three hidden layers having 2000 nodes, 1000 nodes, and 400 nodes, and two output nodes. Having this output layer having two output nodes. So the training of deep learning network was done for over 10,000 epochs for synthetically generated 10,000 mixed pixels. Abundance of soil and urea fertilizer was estimated at the selected wavelength regions. Using a trained deep learning network, abundances of study mixed samples were estimated and compared with the actual abundances. Computed abundances of study mixed samples were used to evaluate the performance of the DASU and DASU alone and DASU based deep learning network. The correlations between the network computed abundance and the actual abundances was analyzed using a linear regression model. So here we have computed the relationship between the DASU alone and the DASU estimated DASU based deep learning network results for the abundances of the fractional abundances of soil and urea. So R square for containing urea fertilizer and silty-clay soil type was 0.325 at 989.3 nanometer wavelength and 0.945 at 1899.2 nanometer wavelength region, and 0.953 at 2195.1 nanometer and 0.643 at 2346.9 nanometer wavelength region. So similarly, R square for the case of the loamy soil. So these results suggest that DASU-based deep learning network computed abundances for all the end members yield negligible variation at 1899.2 nanometer for urea fertilizer and 2195.1 nanometer for both soil types as compared with the actual abundances. So DASU-based deep learning network computed abundances at 989.3 nanometer for urea fertilizer and 2346 nanometer for both soil types held relatively higher differences as compared to the actual abundances. So based on this observation, we have found that I have found that these two wavelength region, that is gives better results, and abundances for these two soil types provided better results at 2195.1 nanometer as compared to the 2346.9 nanometer. And R square results for DASU based deep learning network computed abundances as compared to the DASU alone suggest that later approach is more efficient as compared to the former approach. So means DASU based deep learning network results will have get higher accuracy as compared to the DASU alone results. So based on the box plot results, I have also evaluated the relative error, and in box plot also we have found that these two wavelengths are suitable for the identified the mixed pixel analysis of the urea and the soil sample. So based on our results, these two wavelength regions in this is for the identification of the urea, and this is for the identification of the soil is found suitable to identify the mixed pixels analysis. And these are the results which I have calculated using the DASU estimated abundances, and then next one is for the DASU based abundances for each soil samples. So these are the results in the table. This is the two abundances, and these are the estimated abundances using the DASU method, and these are the errors which I have calculated subtracting the two abundances minus estimated abundances for each mixed samples in two wavelength region. So this is the for second type of loamy soil sample, and same this is the result for the clay soil type sample with urea mixture, and this is the DASU based. This is the result for the DASU based deep learning network for the loamy soil type, and then chemical analysis for silty clay soil, urea fertilizer, and 4 mixed study samples was carried out using the well-known ion chromatography technique in the laboratory calculated 46.6% nitrogen for gram per 100 gram sample in the study urea fertilizer. Measure of N available in study samples was examined individually. Also, the proportion of N in a mixed sample was estimated using conversion factors. Based on the results of chemical analysis, it is found that performances for estimation of fractional abundances of compositions of two soil types and urea fertilizer are well as 2195.1 nanometer wavelength region. And these are the result of the chemical analysis method which I have used, which I have tested using 4 mixed samples, which having 90% soil and 2% urea, 92% soil and 8% urea, 88% soil and 12% urea. And these are the results of the for 2 wavelength regions. So the conclusion, abundances of salt-affected sandy soil have been quantitatively estimated through non-destructive methods using hyperspectral imaging technique. Abundances of compositions of 3 types of soil, silt clay, sandy, and loamy, and NPK compost have been quantitatively estimated through non-destructive methods using hyperspectral imaging. Abundances of compositions of 2 types of soil, silt clay, and loamy, and urea fertilizer have been quantitatively estimated through non-destructive methods using hyperspectral imaging technique. Successfully estimated the abundances of salt-affected soil and micronutrients in different types of soil. Computation of these abundances is relevant to the analysis of soil fertility status for precision agriculture in hyperspectral imagery, with comparisons of in situ hyperspectral data and related spectral library data acquisition acquired through spectrophotometer. Farmers can analyze itself the fertility status of soil without any help of soil scientists. The salient contribution of research work, assessment of abundances of salt-affected soil and micronutrients in different types of soil through non-destructive methods using hyperspectral data, introduced a novel approach for estimation of abundances of salt-affected soil and micronutrients in different types of soil. For future scope, development of the proposed approach may be applied and validated for the hyperspectral field data as a step towards the precision agriculture. So this is from my side. Thanks for your attention.